Hello and welcome. I'm your host, Neil Howard. Thank you so much for returning for another segment. In this segment, we'll be speaking with Dr. George Sledge, Jr. He's joining us to discuss why he made the move to Caris and the groundbreaking work that he'll be doing. Welcome to Health Professional Radio, Dr. Sledge.
Well, thanks for having me, Neil. Great to be here.
Well, tell us a bit about yourself. I did mention that you're going to be doing some groundbreaking work at Caris Life Sciences. Tell us a bit about yourself and what led you to Caris.
Sure. By background, I'm a medical oncologist. I'm a breast cancer doctor, and I've been doing breast cancer research for several decades. Most recently at Stanford University for the past decade, where I served as chief of the Division of Oncology. More recently, I came to Caris a little over 4 months ago. I moved over to Caris for a pretty simple reason. Caris Life Sciences, in its basic business model, is that it does whole exome and whole transcriptomes on patients with cancer, primarily metastatic breast cancer. Caris is sitting on a very, very large amount of data from these whole exomes and whole transcriptomes. The availability of data allows us to do great things. So it was my excitement surrounding this that motivated me to move over.
What is Caris Life Sciences? I mean, are we involved in testing? Are we involved in research, drug development?
So the answer to all of those questions is yes. We start as a molecular diagnostics company, and that is to say we take a piece of a patient's cancer, we grind it up, we measure all of the mutations at the DNA and RNA levels in that cancer, and that allows us to provide information to doctors and patients about the patient's cancer and allows us to direct therapies for those patients. So at a very basic level, what we're doing is helping to provide care for patients with cancer, primarily patients with advanced cancer. But we're also doing many other things. We're involved in fairly high-level research, increasingly using modern artificial intelligence methods to be able to interrogate the cancer at a much, much deeper level, to get some better sense of what's driving human cancers and hopefully some better sense about how we might be able to thwart that. More recently, we've become actively engaged in terms of trying to use the knowledge gained about these cancers to help develop new information regarding molecular targets in specific cancers. Cancer isn't a single disease. It's a broad collection of diseases. These diseases range from the very common to the inscrutably rare. Having the sort of large data sets that Caris has allows us to interrogate each of those cancers, allows us to ask questions about what's driving those cancers, and allows us to see whether or not there are potential therapeutic targets in those cancers that we might have not been able to see in ages past. That's a very exciting prospect for those of us who want to come up with victories over cancer so that we can help patients with the disease.
How much does your molecular profiling differentiate itself from others that are on the market attempting to do the same thing?
Well, we're doing several different things, I would say. In our basic Caris tissue analyses, we measure both whole exome and whole transcriptome. That is to say, basically look at all 23,000 genes that make up a human's genetic data set at both the DNA level and at the RNA level. Now, many of the tests that are out there are only DNA, and many of the tests that are out there only look at a limited gene set rather than whole exome and whole transcriptome. Caris made the decision several years ago that to maximize our understanding of cancer, we needed more data. And as the technology that allows us to do this has become cheaper and more efficient, we have felt that the most data that we can get out of a patient's cancer, the greater the likelihood is that we'll be able to provide useful information to the patient that will stay ahead of the curve and that will be able to learn new things. For instance, if one looks at the Food and Drug Administration's website, one sees approximately one new indication per week on that FDA website occurring over several years. So if one looks at a test that has a limited number of DNA mutations that one's looking at, those tests are more or less automatically out of date within weeks or months and have to be continuously updated. We have future-proofed this by virtue of having whole exome and whole transcriptome. So we can go back if, for instance, there's a new mutation and a new drug attached to that mutation. We can go back and look from something that we obtained a year or two ago, find that mutation, identify it in the patient's tumor, and then tell the patient's physician that it's there, so that will allow that doc to go ahead and treat the patient with a new drug that's just been FDA approved. So we think that's an important part of what we're doing. Now, in the near future, we'll be launching our Caris Assure assay. This is a liquid biopsy, and again, one which differs from those currently on the market in that it will be also whole exome and whole transcriptome off the plasma, but off of the buffy coat, the collection of white blood cells in the blood. We'll be able to look at host DNA in terms of whether or not one has inherited mutations, and we'll be able to look at what's called clonal hematopoiesis, the sort of mutations that occur as a result of aging that we can measure in the blood that are not cancer themselves, but which can look kind of scary, and therefore you need to subtract them as sort of a background contaminant. We think this process of allowing us to do all of these things at once off of a single sample will provide doctors with the greatest amount of information possible, and so we're very excited about it.
What do you think are some of the major challenges that pose a threat to the acceleration of precision oncology?
I think the greatest challenge of all is one that I've already mentioned, which is the problem of one new indication per week. Now imagine you are a general medical oncologist taking care of patients, and imagine that every single week of your life you have to learn some new indication. And by indication, I mean the use of a drug for a particular disease. Every single week, if you don't have a way of keeping up, you're getting further and further behind. Over the course of several years, you're now talking about potentially a few hundred indications. So this educational burden for patients is immense. The financial burden on patients and the system is immense. The burden on healthcare systems, on insurers, on federal payers is immense, and it's only going to get greater and greater and greater. So the big challenge in this area of precision oncology, the big challenge is going to be to make sure that doctors and patients are adequately educated in terms of the best way to use this data, and that payers and the government and other stakeholders have a good sense of what's going on. I think this is a huge challenge for all of us.
Is there anything that you'd like to add for our listeners? And then give us a website where we can learn more about Caris Life Sciences. CarisLifeSciences.com is a good place to reach us. In terms of what else I'd like you to know, I think the next decade is going to be perhaps the most exciting time in the history of oncology. We're going to learn more and more about what makes these evil cancers tick. We're going to find better and better ways to combat them. Hopefully we'll get closer and closer to a time where we'll actually be able to be serious about thinking about how to prevent them from occurring. And ultimately this hopefully will transfer to longer lives for patients. And one more time, if you would, that website for us.
CarisLifeSciences.com.
George, I appreciate you lending us some of your time this evening. It's been a pleasure speaking with you, and I'm looking forward to our next conversation.
My great pleasure, and thanks for having me.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. George Sledge. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes. Listen in, download it, Anchor, Spotify, and be sure to subscribe to our YouTube channel at youtube.com/Health Professional Radio.